Hello everyone. Let's get started. Today we're going to be discussing the regulation of gene expression in prokaryotic microbes. The way we're going to approach this topic is to break it down into three sections. We'll talk first about why bacteria and archaea bother to regulate gene expression. We'll talk about where in the pathway from DNA to active protein genes are regulated. And then we'll talk about exactly how genes are regulated. And there are three mechanisms that we will touch on. The first is the regulation of the state of the chromatin. Second is the use of proteins called transcription factors. And third is the use of activator and repressor proteins to regulate gene expression. And we will use two common examples: maltose regulation and arginine regulation. So the first question is why? Why would a microbial cell bother to invest time and energy into carefully regulating when genes get expressed? There are really two reasons that a cell would do this. First and foremost is to conserve energy. Remember, microbial cells, in fact all cells, are very thrifty. They're going to try to conserve energy wherever they can. Nothing, really, should be wasted in a living cell. So because the expression of genes is expensive to a cell, it costs things, it costs energy, it costs enzymes and substrate molecules. They're only going to do this process of gene expression when they absolutely need to. The second reason that microbial cells bother to regulate their genes is that they must be able to rapidly respond to changes in their environment. As we've discussed before, microbial cells are quite different from multicellular organisms in that they are exposed to a lot of extreme conditions in the environment. We talked, for example, about temperature changes and pH changes. A cell like this has to be able to rapidly alter the kinds of proteins that are being produced in order to respond to those kinds of changes. Now I'm going to introduce a new term here, which we apply when we're talking about the regulation of gene expression, and that's this word constitutive. Some genes inside of cells code for proteins that cells have a need for on an ongoing basis. In other words, there are some proteins that a cell needs all the time, and it needs to continually make those proteins around the clock. Sometimes we refer to these as housekeeping proteins. These genes that code for these housekeeping proteins therefore must be expressed 24 hours a day, 7 days a week. These kinds of genes that are expressed all the time are genes that never really need to be regulated. We just need them to stay on so that the protein gets made constantly. This kind of gene expression is called constitutive gene expression. Other genes are coding for proteins that a cell only needs periodically. Proteins that a cell has a need for at a particular time of the year, or a particular stage of life, or under particular environmental conditions. These genes you would not want to be expressed constantly around the clock because these proteins are not needed around the clock. These are the genes that are under regulated gene expression, and actually most genes are under regulated gene expression. So what do we mean when we say that a microbial cell is regulating gene expression? We're talking about the cell taking control over if a protein gets made, when that protein gets made, and how much of that protein gets made. When we think about the central dogma, we can see that there are lots of opportunities for a microbial cell to take control of protein production. It can take control anywhere from the time of transcription all the way to the time of folding up and activating a protein. We talk about this pathway of the flow of information in a cell. Let's recall the central dogma. It tells us that DNA is used to make RNA through the process called transcription. So certainly a cell could take control over whether or not a gene gets transcribed. And in fact, that's a very common place for a cell to step in and regulate gene expression during the time of transcription. The dogma then tells us that RNA is used to make proteins. And there are actually a few places where a cell could step in here. This is the time of translation. And as we'll see in a minute, there are different opportunities during translation and during the time that a newly formed protein gets folded up and activated, where a cell can step in and determine whether or not that process should be allowed to continue. So, regulating transcription, regulating translation, and in fact, regulating the newly made protein are three examples of where a cell can step in and regulate or determine whether or not we should keep going with gene expression. Let's think about the regulation of transcription for a minute. The most common way that a cell regulates the transcription of a gene is through what we call negative control. In other words, for most genes that are under regulation, the default condition is that transcription is on, that transcription is happening. Another way we describe this is by saying that transcription is up-regulated. We're turning it on, like a light switch. It's going up, it's going on. Now, when we step in, or when a cell steps in with control mechanisms, it would then turn transcription off, or what we more commonly describe as down-regulating transcription. So most of the time, a regulated gene is in this default position of, yeah, let's go ahead and transcribe this gene. A cell has to take the step of bringing that transcription level back down, either turning it off or down regulating it, making it happen less often. The second place where genes are often regulated is during the process of translation. So, in other words, sometimes transcription occurs and we make an RNA from the DNA, but then the cell determines that we don't need to translate that protein right now. We don't need to actually produce the product right now. So we can step in at that point and stop this process of gene expression. One of the ways that this is achieved is by taking that newly made RNA, the messenger RNA that was produced during transcription, and just degrading it. Cells can, in other words, determine whether or not an mRNA is going to get translated They can even step in during translation while mRNA is being translated and degrade the mRNA so that translation stops at that point. Remember, a single mRNA is going to get translated over and over again. In other words, the cell is going to make several copies of protein from that one mRNA. It will keep translating it as long as the mRNA exists. So a cell can degrade the mRNA in order to reduce how much protein gets made. The other thing a cell can do, instead of just degrading the mRNA outright, is to use a molecule called a small interfering RNA, or what we shorthand as siRNA. This is an RNA that cells produce that is complementary to the mRNA that would be produced during the transcription of a gene. Small interfering RNAs are regulatory molecules. They can be sent out into a cell and they will find and bind to the mRNA that they match, that they are complementary to. And what you end up with then is a double-stranded molecule of RNA. You've got the mRNA, and then bound to that, you've got one of these siRNAs. That double-stranded molecule of RNA can't be translated. So essentially, by using siRNAs, a cell can very rapidly bind up and inactivate any mRNA that's floating around out there. That will block translation of that mRNA and prevent any more protein from being made from it. Finally, a cell can step in pretty late in the process of gene expression and still get control over protein production. This is regulation of protein activation and inactivation. Remember that at the end of translation, what the cell has produced is a polypeptide, a long strand of amino acids that was coded for by the mRNA. This strand of amino acids is not yet functional. This long chain of amino acids, and remember, sometimes these polypeptides are thousands of amino acids long. This long chain of amino acids has to be processed before it's an active functional protein. That long chain of amino acids has to be folded up into a very intricate three-dimensional molecule that will then be active. So cells can do a couple of things here. They can step in and prevent all of this folding from going on. They can even take a protein that's already been folded up and is ready to start working, and they can inactivate it by doing things like attaching small molecules to it and causing it to change its shape. And of course, the last thing a cell could do is simply degrade a protein that was just made. This is sort of a last-ditch effort because at that point, the cell has already wasted the time and the energy building the protein. So this is sort of a last-ditch way to get rid of a protein that a cell doesn't want to be active in this moment. So there are lots of places in the flow of information from DNA to active protein where a cell can step in and regulate what's going on. Regulate if the protein is being made, when the protein is being made, and how much of the protein is being made. Take a look at this drawing on this slide. Now, this drawing was actually made as a representation of the regulation of gene expression in a eukaryotic cell. And I know that because, as you can see, they've shown us that this side of the diagram is taking place in the nucleus, and this side of the diagram is taking place in the cytoplasm. Obviously, in a prokaryotic cell, we don't have these compartments. All of this is happening out in the cytoplasm. But the diagram is still useful for us because it shows us all of the places between DNA and active protein that a cell can step in and regulate the production of that protein. Number one, of course, is controlling transcription. And this is the most obvious place to step in. By interfering with this process where DNA is used to make RNA, a cell can step in early and effectively and stop the production of a protein. Then they show us that, number two, that a cell could step in and control the processing of the RNA. We didn't talk about RNA processing because it doesn't happen much in prokaryotic cells. So let's skip that one. Let's skip number two. Out here we have RNA transport out of the nucleus. In other words, we can skip that one too, because that doesn't happen in prokaryotic cells. But here's one we can pay attention to, and that's controlling the process of translation. Remember, translation, that's where the mRNA is used to make a protein. We said that microbial cells can step in right away and they can inactivate that mRNA by releasing small interfering RNAs that will bind to it and make it unavailable for translation. Cells can also just literally degrade the mRNA so that it can't be translated. And then down here on the lower right corner, we see the last place where a microbial cell could step in and regulate gene expression, and that's at the level of the protein. When the long chain of amino acids, the polypeptide, is produced in translation, a cell can certainly step in and prevent that chain of amino acids from being folded up into an active protein. A cell could leave a protein in an inactive state, have it available, but inactivate the protein until it has a need for it. So these are all of the places where microbial cells, and in fact other cells, can step in and regulate the expression of their genes.